Heterocyst. Heterocysts are specialized cells that are found in the cyanobacteria or blue-green algae, and they are responsible for the nitrogen fixation in the cyanobacteria. They occur every 5 to 10 cells, in between every 5 to 10 cells in the filament. This is the part 2 of the video. I have made the in the part 1 of the video I have discussed the overview of the heterocyst, its structure, and in this video, the part 2 of the video, I will be discussing the molecular aspects of the heterocyst pattern formation. In case you haven't watched the part 1 of this video, I would request you to watch that video first, because that video also contains many informative things, and after watching that video you can watch this part 2 of the video. Hello friends, I am Ayantika, and I do videos regarding the botany syllabus for MSc BSc, and in case you are new to the channel, subscribe to get the latest update of the videos that I make. So before heterocyst, starting the video on heterocyst, there is a quiz. Which nitrogenase is found in legume diazotrop? Now there are options, and nitrogenase is the enzyme that is responsible for nitrogen fixation. So certain type of bacteria, the diazotropes and cyanobacteria, both these they fix nitrogen, that is they fix atmospheric nitrogen to reduced ammonia form, and during this formation they need nitrogenase enzyme, which consists of two. It is an enzyme complex of two enzymes, dinitrogenase and dinitrogen reductase. And so depending upon the type of metal that is present in the nitrogenase, nitrogenase can be of different type. Now in this question, I've just asked what type of nitrogenase is present in the legume diazotrop, in the that is a bacteria that is present in the root nodules of the leguminous root nodules. That bacteria contains what type of nitrogenase? That is the question. If you know the question, if you know the answer, you can write it in the comment section. But if you don't know, I will be discussing it at the end of the video. So stay with me till the end of the video. The molecular aspect of heterocyst. In the last video, I have given you various aspects of heterocyst, how they are formed, and what are the special, what are the events that that occur from the normal somatic cell to proheterocyst, then the heterocyst, and how senescent what happens to senescent heterocyst. All this I have discussed in the previous video. Now I will be just discussing the molecular aspect, that is the genes and the proteins that will be responsible for the entire molecular the pattern formation of the heterocyst. For the expression, for the formation, conversion of somatic cell to heterocyst, there are almost 45 genes that are responsible. But we are not going to discuss all the genes. We are just going to talk about the genes that are very important. Like for example, HETC, HETF, HETL, HETR, and HRRA, PATA, ASR131735, HENR, HEPA, HEPK, HEPN, HEPS. Now among these, the most important is HETR. We will be seeing that HTR is the most important gene that produces transcription, that actually produces a protein HTR that homodimerizes to bind to the DNA promoter region, and in turn produces various type of proteins that are essential for the conversion of somatic cells to heterocysts. And apart from protein, there are also polysaccharides that are involved in the formation of heterocysts, because we know that the cyanobacteria, just like other gram-negative bacteria, they have a thick polysaccharide wall, but this heterocysts have much more much more thicker wall than the normal somatic cell. So for the thickening of the wall, polysaccharide needed to be synthesized and transported to the wall. So for the polysaccharide synthesis, the expression of genes that are needed are HGLB, HGLC, HGLD, HGLE, and HGDD. And for the transport of this polysaccharide from the site of synthesis, that is the cytoplasm, to the cell wall, they need transport proteins, and these transport proteins are in turn produced by the expression of genes HEPA, HEPB, HEPC, and HGLK. Now, overexpression of HETF and HETC, it will cause a continuous heterocyst formation. So, all these are stimulatory genes that are needed for the heterocyst transformation. Also, for the glycolipid export, that is, we also need DEV, BCA gene and HGD genes. If there is nitrogen deficiency, then only the heterocyst will be produced. Cyanobacteria, just like other type of bacteria, it has operons, that is group of structural genes that are auto-regulated, which means that, say for example, if there is ammonia and nitrate in the atmosphere or in the surrounding environment in which it is growing, then the cyanobacteria will not produce enzymes or proteins that will be needed in the metabolic process that involves nitrogen fixation. So because there is no need for nitrogen fixation when already ammonia salts or nitrate is present in the media. But if there is nitrogen deficiency, then this whole entire for the production of nitrogen, for the reduction of nitrogen into ammonia, heterocysts needed to be formed, because we know that the nitrogenase, all type of nitrogenase, they are sensitive to oxygen, that is, they get damaged by the presence of oxygen. So this in the nitrogen, if there is a nitrogen step down in the media, that is a deficiency in the nitrogen, then that will be detected by a curved cycle intermediate, that is the 2-oxoglutarate. This 2-oxoglutarate, this acts as a transcription factor. This protein 2-oxoglutarate, it acts as a transcription factor that regulate NTCA gene. Now this NTCA gene in turn regulate the HETR gene. HETR gene is very much essential for starting the pattern formation of the from the somatic cell to the heterocyst formation. And also in not only in absence of nitrogen deficiency, we see oxoglutarate accumulating in the cell, but also we also see another thing, that there is also very high amount of calcium. in case of nitrogen deprivation. This high amount of calcium is because of inactivation of another gene, that is the CBP. So this high amount of calcium, it is also a character, it is also an indicator that there is less amount of nitrogen in the media. And there is also other transcription factors, such as sigma C. These are the transcription regulators that stimulate that they stimulate the heterocyst formation during the early stage. And the sigma E are the transcription regulators that are expressed in the late stage of heterocyst development. And sigma G is an inhibitory transcription regulator. That is, sigma G, this protein sigma G will inhibit heterocyst formation. And the earlier two proteins, that is a sigma C and sigma E, these transcription regulators, they are stimulatory, and sigma G is inhibitory. So PAT-S is another gene that has inhibitory effect on heterocyst differentiation. That is, when the gene PAT-S it expresses, then the PAT-S protein, it causes the suppression of heterocyst. Earlier we saw in the previous slide also we saw that sigma G was responsible for heterocyst differentiation suppression, and here we see there is PAT-S. Other such proteins are PAT-A and CBP. All these, they downregulate or suppress the conversion of vegetative cell to heterocyst. Another inhibitor is HCTN. And for the nitrogen fixation, we know that heterocysts have developed various mechanisms so that the oxygen do not enter, and just for the formation of the nitrogenase enzyme, we need genes from the NIF operon. Now, nitrogenase enzyme is an enzyme complex with two enzymes. The dinitrogen reductase, what it does is it transfers electron from ferredoxin or flavodoxin to the dinitrogenase, and dinitrogenase, it reduces the dinitrogen, that is the atmospheric nitrogen, to ammonia. Now, there are different versions of nitrogenase enzyme depending upon the type of metal cofactors, the metals, they are present in the structure, and different type of nitrogenases have a requirement of different metals as cofactor. And the main NIF operon genes that are responsible are NIFB, NIFE, NIFH, NIFN, NIFX, NIFW, NIFU, and NIFS. These are the main genes that are responsible for the formation of various nitrogenase enzymes. And these genes, they are not expressed in the first half of, that is in the early heterocyst formation, but they are formed by the rearrangement of they are actually they are stimulated for they are stimulated for expression, that is only after heterocyst formation is complete, that is in the later stage of molecular pattern formation for the heterocyst production. These genes are expressed. And all these genes, that is the genes that are required for heterocyst formation, genes that are required for heterocyst maintenance, and genes that are required for the nitrogen fixation, that is a form for the formation of nitrogenase, all these three categories of genes are conserved in all the cyanobacteria species. Now is the quiz time. So I have been discussing so many times that nitrogenases have various versions depending upon the metal ions they contain in their structure. So among the three nitrogenases, which nitrogenase is found in the legume diazotroph? Diazotrophs are actually the bacteria that are present in the leguminous plants, and these diazotroph bacteria, they fixes atmospheric nitrogen. So among the options, molybdenum nitrogenase are used by the legume diazotroph to fix nitrogen. So the option will be first option. Now here I should be also saying that cyanobacteria have all these three type of nitrogenases depending upon the type of cyanobacteria we are talking about. With this, I come to the end of the video. I have made many other videos regarding the cyanobacteria, and I have also made videos regarding algae lichens. So I have put the link, all the links to the my previous video in the description box. You can, if you want to watch those videos, you can just click on to the link to watch the videos. Thanks for watching this video. Please like it and share it with your friends, and if you subscribe to this channel, will be encouraged. And also, if you want me to make any videos regarding any topic, you can also suggest it in the comment section. Thanks for watching this video.